In this video, we're going to be talking about insulin, and we're going to be answering a few questions about it. So first of all, what is insulin? Well, it's a hormone which is released when blood glucose levels are high, and it plays a major role in energy metabolism and in homeostasis. It does this by binding to tyrosine kinase receptors that will phosphorylate substrates within the cell until a protein called AKT is activated. AKT will go to promote the translocation of GLUT transporters onto the membrane of the cell so that sugars can enter the cell and be stored and used. Insulin is produced in the beta cells of the pancreas. Insulin's sister hormone, glucagon, is produced by the alpha cells in this same organ. So what is diabetes? Diabetes is a disease that is separated into two different types. Type 1 diabetes, which is an autoimmune disease where the beta cells in the pancreas are destroyed and patients can't produce enough insulin. So these patients will need regular insulin administration, especially after meals. In type 2 diabetes, the body can't produce enough insulin, and this is often due to insulin resistance. And so patients with type 2 diabetes need to avoid sources of dietary sugar so that they can limit the need for long-term medication. So how do we produce artificial insulin used in type 1 diabetes treatment? So we do it via recombinant DNA. In recombinant DNA, we take the gene that codes for insulin and we take a plasmid, which is bacterial DNA, and we cut this plasmid with restriction enzymes. We then insert the gene that codes for insulin into the plasmid, and that bacteria now becomes a recombinant bacteria. We let the recombinant bacteria ferment, and we harvest the insulin that it produces. I hope you found this useful, and thank you for watching.